or you can study it by next generation sequencing. In fact, we recently obtained the next generation sequencer. Before that, for a decade or maybe 2 decades, we have been culturing, and then recently we did real-time PCR. So what is this real-time PCR? In real-time PCR, what you do is that you do the sequencing of candidate bacterial gene. Let us say there are 1,000 species of bacteria in the gut. If I have to do 1,000 species, you can understand how many real-time PCR I have to do. So per patient, I have to do 1,000 PCR per sample. And then if I take 100 patients, you can say 100 into 1,000, so difficult. Also, real-time PCR means we use dilution so that I can quantitate, so that you can get a quantity of the bacteria, not necessarily presence or absence. And second thing is, typically what we do is that you take a standard bacterial species. For example, we use in our study ATCC strain. ATCC is American type culture collection, but there is also Indian culture collection in Pune. So you obtain the bacteria from them, compare with the sequence. Also there you do a dilution and compare the carbs, so by melting carb analysis, that what is the quantity in relation to ATCC strain that it took. So this is a real-time sequencing. But there we'll say I'll study 4 candidate bacteria which I know to be likely to be good bacteria. I'll study 4 bad bacteria and look at the ratios. We are not studying 1,000 species. We are studying only 8 species because that is what is doable. But now what we do is this next generation sequencing. What is the beauty of next generation sequencing? It can do all the PCR at a time. Like when you do Sanger sequencing, you use ddNTP so that you do a chain termination, then you use a fluorescent dye to label that chain, and then you find out the sequence. But here you don't have to do that. What you do is that you take this chip. This chip has maybe 5 million well, 520 million well, 540 million well, and so on. So this is CMOS technology, by which you have a huge number of well. Each bacterial DNA will get into one well. Okay, each bacterial DNA will get into one well. And then what this system does is, it throws the bases one after another. So typically it will throw adenine. So once it throws adenine, what will happen? That if a particular well has thymine, so adenine will combine with thymine, and this combination will release hydrogen ion. Now the moment it releases hydrogen ion, the pH of this well will change, it will go down, and the system will capture that this particular well has thymine, because after throwing adenine, its pH changed. But on the other hand, another well where there is no thymine, pH will not change, so system will actually pick up where adenine is there and where guanine is there, and one after another the system throws the bases. You know, understand? So here you can study thousands of species in one go, and you know the bacteria in a very wholesome manner. Now these are the terminologies now people are saying. Like when you talk about India, what do you say? What we say is unity within the diversity. So if you are diverse still united, it is better. Similarly in bacteria, if you are diverse, you are better. If you are not diverse, you are bad. We talk about alpha diversity. What is alpha diversity? It is an intra-sample diversity. That means if I take the fecal material of a person X, and if I get bacteria which are very same, that means this fellow has very low alpha diversity. So he is likely to have a disease, because diversity greater is better. Whereas another person, let us say, look at this person, he also has quite different bacteria than this person, but still he has low alpha diversity. You know why? Because the bacteria is not diverse. But compare these two individuals. This is a high beta diversity. That means inter-individual variation is beta diversity, intra-individual variation is alpha diversity. Like here, this fellow has high alpha diversity, and diversity greater is better. So in bioinformatics, they use Shannon index, Simpson index for alpha diversity, Sorensen index, Jacquard index for beta diversity, and so on. Now remember only one thing, that diversity is better. What is small intestinal bacterial overgrowth? Small intestinal bacterial overgrowth is a situation where there is excess bacteria. For example, I told you that the jejunum or upper gut is supposed to have at most 10 to the power 3 colony forming unit per milliliter of the aspirate. But suppose this goes above 10 to the power 5, this is excess bacteria, and the excess bacteria means it will compete with the food that we are eating, so the fellow may become malnourished, the fellow will lose weight, and because the food is fermented by bacteria, the fellow will have a lot of gas, the gas will distend the gut, and therefore he will have pain, he may have diarrhea because of osmotic load. So there are different disease conditions where we find that there is bacterial overgrowth, but because these are technical, I will not talk about. Now, I'll not talk about this. This is one of the study that we did, took 8 bacterial species, because we are doing real-time PCR. There is a disease called irritable bowel syndrome, which you would not understand, need not understand. It is a chronic GI disease. And we took healthy people, took IBS patient, looked at, is there good diversity or is there bad diversity? Is the bacteria different in IBS versus healthy control? And what we found is very interesting. If you get a Pseudomonas in the gut, it is a bad bacteria. If you get a Pseudomonas in the gut, it is a bad bacteria. You know why? Because Pseudomonas produce some toxin. This toxin targets the nerves of the gut, and the nerves of the gut gets or become malfunctional, so that gut movement changes, and that leads to disease. So this study showed Pseudomonas is a bad bacteria. You should not have it in the gut. We are talking in this meeting about microbe-human interaction. Let us say I tell a fellow, how are you? He warmly replies that I am well, how are you? This is a behavior. Another fellow I ask, how are you? He says, who are you to ask me that how am I? Are you known to me? This is a different reaction. But the question was same. Similarly, one fellow can get a bacteria and his immune system can talk differently, very politely. Therefore, he will not have inflammation. He will not have disease. Another fellow can have an immune system which is too aggressive and will react with inflammation with so-called normal bacteria and will develop disease. So therefore, remember, Robert Koch said it is all the organism which causes disease. He was wrong. I tell you, he was wrong. Now some of these physicians would say it is all in the human gene that causes disease. They are wrong except for Mendelian diseases. Okay? It is the host microbiome interaction which decide whether I will be diseased or I will not be diseased, and it is very nice that we are talking about a meeting of host microbiome interaction. Really, if you want good quality research, work on host microbiome interaction. I'll show you an example. This is a disease called irritable bowel syndrome. We took 209 such patients, 273 healthy people, and we did genotyping of interleukin-1 RA gene. So interleukin-1 is a pro-inflammatory cytokine. Wherever there is interleukin-1, there will be inflammation. So obviously it causes inflammation because it occupies a receptor and then do a signal transduction. Now what happens is there is a receptor, therefore there is a receptor antagonist. If that antagonist is occupying the receptor, though interleukin-1 is liberated, but it can't cause inflammation. So what we looked at is that what is the interleukin-RN gene polymorphism among healthy and the disease. And what we found is yes, those people who are diseased, they have a genetic polymorphism which is associated with the less expression of interleukin-1 receptor antagonist. That means they are pro-inflammatory type people because their gene is underexpressed, that receptor antagonist gene. Whereas the healthy people, their gene is optimally expressed. That's why they will have less inflammation. To see that further, we looked at the gut microbiota by jejunal aspirate culture. We got some people who had bacterial overgrowth, some people did not have bacterial overgrowth, and we looked at interleukin-1 alpha and beta level in the mucosal biopsy, and what we found is those who have bacterial overgrowth have more interleukin-1 alpha and beta than those who did not have bacterial overgrowth, and also it correlated with the genetic polymorphism. So this is one gene study or one cytokine study for a pro-inflammatory state and gut microbiota host interaction. To study it further, recently we took up this study where we took up a lot of cytokines which are either pro-inflammatory or anti-inflammatory. For example, interleukin 6 is a pro-inflammatory, interleukin 10 is an anti-inflammatory, CXCL11 is pro-inflammatory chemokine, CXCR3 is a pro-inflammatory chemokine, and also we look at TLR. So what are TLR? TLR are toll-like receptors. It is like, let us say, I know a fellow, I know Dr. Talukdar. How do I know Dr. Talukdar? I have developed, my brain has developed a pattern recognition that he looks like this. Before I knew him, my brain has no idea if the fellow looks like this fellow will be Talukdar. Okay, no. But with repeated visit, I developed a pattern recognition in my brain. Similarly, TLR are pattern recognition molecule in our gut. They actually looked at the pattern of the bacteria. So this is called pathogen associated molecular pattern. Then if they find some bacteria has a pathogen associated molecular pattern, they will incite the immune response. Whereas if they find this bacteria doesn't have a pathogen associated molecular pattern, no immune response is initiated. Okay? Second thing also, they recognize damage associated molecular pattern called DAMP. So if a bacteria has damaged a tissue, that will also be recognized. And we looked at this. We looked at the pattern recognition receptor. These are TLR. We looked at the chemokine. We looked at the cytokine in relation to the gut microbiota. And we found, yes, this is the story. It is not in the microbe alone. It is also in the host response. Now, next issue is we talk about undernutrition. We say the people in the West are obese and the people in the East are very sort of malnourished. And we thought everything is related to calorie availability. That means the fellow who has more calorie, he becomes obese. But look at the elephant. Elephant is very obese, but he takes a food which is very low calorie. Okay? Look at the lion or tiger. He takes a very high calorie food full of fat and protein, but he's very lean. So it is all related to calorie availability? No, I'll show you some data. Look at this. Okay? Look at this. This is a study which is Bill Gates Foundation funded study where there are 8 countries study, and they found a particular type of gut bacteria, pathogenic bacteria you have in your gut becomes leaky. Because you have increased intestinal permeability. Also, you keep on losing calorie. The one side, because of the economy, you have less calorie available. Second side, you have microbes in your gut because of your poor environment. Like in Bangladesh, you go near the coastal side in the village, you see the environment. So they develop pathogenic bacteria in their gut and they keep on losing calorie. On the other hand, you can have calorie extracting bacteria like elephant has. They will extract calorie from the most low calorie food and will cause weight gain. Now you want further evidence? This is very interesting. Scientists discovered this and published this in 2012. But much before that, 50 years, 60 years, 80 years in animal husbandry, if you go to animal husbandry and ask them, what do you do? They will give low-dose antibiotic to animal from their childhood. Why? Because they know that if you give low-dose antibiotic to the animal, he has more adipose tissue, more protein, and therefore he will give more meat. So this experiment was done just in 2012 where these mice, they were fed antibiotic or water and they looked at their microbiota and the weight gain and the gonadal fat and the body fat and the protein mass, and they found that giving low dose antibiotic lead to weight gain. That means manipulation of gut microbiota can change weight. Either you can lose weight by having some bacteria which makes your gut leaky. They are not calorie extracting bacteria. On the other hand, if you give bacteria on the other side, you can gain weight. Now this is a study in human. This was an UK study which showed that those children who received antibiotic for something like respiratory tract infection or GI infection in UK are the one who became obese. So giving antibiotic leads to obesity. Look at further. This is a very interesting study. What they did is they took mice And to the mice, obviously this is a germ-free mice, I think, genobiotic mice. Now what they did is, these mice, they fed high carbohydrate diet and high protein diet. They fed the mice high carbohydrate diet and high protein diet. Now you have some bacteria, for example firmicutes, they actually, or saccharolytic microbes, they like to eat the carbohydrate. Like you have a lot of people who are vegetarian, you give them non-veg food, I can't take this, they will rather starve themselves. Okay, similarly the bacteria, they are saccharolytic, you give them protein, lot of protein, they will not eat. So what will happen if they don't eat? They will die. They will die. Some of them will say mucus has carbohydrate, so they will actually start eating the mucus. They will degrade the mucus. The mucus is the first line of defense in our gut. So this experiment proved that giving dietary fiber led to more saccharolytic microbiota, giving protein led to more proteolytic microbiota, then giving protein led to degradation of mucus layer, and the mucus layer degradation, when they challenge them with pathogenic bacteria at a much lesser dose of bacteria, these mice got infected. What does it mean? That mucus layer is the first line defense. So too much animal food is bad. Too much animal protein is bad because it promotes the growth of proteolytic microbiota. Now the whole world is working on it. Like go to the US, they have tremendous frequency of colon cancer. Whereas come to the India, frequency of colon cancer is very less. It may be in their diet. Go to the Western Mall, you will find the vegetable is in one corner and they have a lot of meat, and these meat are not freshly sort of cut meat. These are preserved meat. So now there are a lot of studies coming up. What does food preservative, particularly meat preservative, do to the gut microbiota? They lead to lethal action on the gut microbiota.